Hello, everyone. Welcome to our webinar today on applications of flow and imaging cytometry to COVID-19 research. My name is Kamala Tyagarajan, and I'm Director of Flow Cytometry Assays and Applications at Luminex Corporation. During this presentation, my colleague, Dr. Haley Pugsley, and I will share how our flow cytometry and imaging technologies at Luminex can be utilized in aspects of COVID-19 research. The rapid spread of COVID-19 has created an acute scientific need to understand clinical manifestations and mechanisms of the disease, and also understanding how to monitor and tackle the disease. Intense research is ongoing worldwide at the molecular, protein, and cellular levels, and Luminex provides a variety of solutions in all these approaches. In this presentation, we will mainly focus on COVID-19 research enabled by cytometry. So major questions in COVID-19 research by cytometry can span: how does the virus impact the immune system? How does the immune system respond to the virus? Viral entry mechanism, and what drug or strategies can be identified to modulate the virus. If we look at areas of viral impact on the immune system, numerous studies are being published on what is ongoing with the immune cells in different stages of infection, and if they can provide clues and predictors about the infection as to how mild or severe the disease is. Select few examples are indicated on the right, and some of these early studies have found some key markers. Many have identified a pattern where the T cell absolute counts, the CD4 and the CD8 counts in blood from COVID-19 patients are drastically diminished in response to severe infection. And further, some of these populations also seem to recover as the patient recovers. So phenotyping of blood samples with regard to absolute counts and percentage is critical. And in many cases, people are performing TB, NK lymphocyte analysis, as well as evaluating T cell differentiation into naive and memory subtypes. Many studies are also identifying markers of inflammation. For example, inflammatory monocytes have significant morphological and functional changes with COVID-19 infection and have been shown to be involved in progression of the disease. Several studies are demonstrating the importance of T cell exhaustion markers such as TIM3 and PD1, as well as increased CD8 expression and T cell activation markers. Along with impacts on the immune cell populations, people are also studying the immune response mechanisms, and several very interesting publications of late have shown study details where PBMCs are stimulated with viral antigens, and the consequent impact on the immune cells are being evaluated. These kind of studies not only characterize the immune cell response, but also look at secreted cytokines, and intracellular cytokine measurements are also being performed. And these kind of approaches are, again, very important in vaccine development strategies as well as drug development approaches. Another big area of research is studies evaluating viral entry and infection mechanisms. People are interested in how the virus is interacting with certain cell types. They are creating in vitro models to understand the viral mode of entry and infection, to understand what kind of protein and expression markers are involved, and how these are impacted by viral binding and infection. And drug screening and repurposing studies are also being extensively performed to identify effective antiviral strategies where people are taking existing approved drugs and then applying them to viral infectivity studies, and several papers discuss in these approaches as shown on the right. At Luminex, we offer a range of platforms and solutions that can enable cytometry and provide information in COVID-19 research. These include our platforms based on microcapillary cytometry, such as the Guava Muse Cell Analyzer and the Guava EasyCyte platforms as shown on the left, and our range of imaging flow cytometry platforms, which combine the power of imaging along with flow cytometry and can provide unique insights into studies such as inflammatory mechanisms of COVID-19. During this talk, I will be mainly focusing on microcapillary flow cytometry, and my colleague, Dr. Haley Pugsley, will discuss imaging flow cytometry. So let's first take a look at what we mean by microcapillary cytometry. Microcapillary cytometry, which is shown on your left, differs from traditional flow cytometry on the right, shown on your right, that in that it does not utilize sheath fluid, and instead the sample is aspirated directly from a tube or a valve through a microcapillary flow cell and interrogated by a laser, and data is collected from detection channels as it flows through. This simplification of fluidics results in a more compact system, and these which are easy to operate and need minimal maintenance. Microcapillary cytometry gives rise to some distinct advantages. First, because of the nature of the technology and the use of a precision pump, we can get absolute cell count information without having to add external beads. And this capability to get absolute counts automatically brings benefits in many application types. The design leads to small cellular samples being needed for analysis, and this can be very valuable in COVID-19 research, where researchers are trying to maximize the information from precious samples available. The lack of sheath fluid also leads to low biohazardous waste generation, which is again highly advantageous when dealing with infectious samples. So let's take a closer look at our two platforms based on microcapillary cytometry, the Guava Muse system and the Guava EasyCyte system, both of which are based on microcapillary cytometry. So the Guava Muse Cell Analyzer is an easy-to-use, simple, and compact instrument for cell analysis. The instrument uses a 532 nanometer laser and provides information based on forward scatter and two fluorescence detection channels. The platform has a very small footprint and is portable and can easily fit anywhere, even in the biological hood. And this is an aspect that can be very helpful as studies are performed on COVID-19 samples, where people might want the instruments inside the hood or proximal to it. The platform is based on microcapillary cytometry that we just discussed, and this provides the advantages of absolute counts and percentages, as well as needing small samples and generating low waste. One of the key features of the instrument is the intuitive touchscreen interface, which guides the user through the steps and makes the acquisition and analysis very easy. There are a whole range of dedicated kits for the platform, along with assay-specific software modules, which allow a variety of experiments to be performed and quantitative cellular data to be obtained at the single cell level. There's also an open module software module available on the platform, which is allows the user great flexibility in terms of the experiments that they want to perform. Kits on the platform range from areas of cell health, immunology, to cell signaling, and thus spanning broad application areas. So COVID-19 research can often require quick screening and characterization of blood and PBMC samples. So the assays of the Muse, which can be really valuable in this kind of work, are assays such as Muse count and viability assay and the Muse immunology assays. Both these assay types are very simple to perform, no-wash assays, and using dedicated reagent sets, which have been well validated, you can quickly obtain information on PBMCs or blood samples. So let's take a closer look at the Muse count and viability assay. This is a very useful assay for determining cellular concentrations and viability status in a cellular sample, and this assay has been extensively applied to a wide variety of cell lines, and many studies have been performed comparing this assay with different methods. The assay is accompanied by a dedicated software module and can provide summarized data outputs as shown on the left in an easy format for the user to deduce the results and the next steps. In the slide, we show examples of applications of the assays to the study of PBMCs. As shown on the right plot, we can really obtain a very good wide measurement range with a nice linear response with either fresh or frozen PBMC samples. The assay provides for very good precision, and a low percent CV is achievable for both count and viability results as shown on the table on your left. So the assay can thus provide very important viability or concentration assessment of PBMC samples, which is important if you're planning on immune response studies or evaluating status of your sample or pre-screening your samples for a broader study. The Muse immunology assays are powerful assays that provide a lot of very valuable information on T and B cells in blood or PBMC samples. There are specific separate assays for CD4 T cells, CD8 T cells, and B cell determination, and each assay provides absolute counts of the populations, total lymphocyte counts, and population percentages based on total lymphocytes. The assays employ no-wash protocols, and the easy-to-use instrument delivers accurate, reliable results. This combination of the protocol as well as analysis allows for very high precision, as can be seen on the data table on the right, and where in inter-operator studies, very low percent CVs were obtained across multiple operators on multiple instruments analyzing the same samples. So we've also compared the results obtained from the Muse immunology kits with other platforms. And in this example, we're showing you the results from the Muse CD4 T cell assay. The results obtained on the Muse for a wide variety of whole blood samples was compared against results from a higher-end flow cytometer and kit. And what we see based on this data set is that we are really getting very nice correspondences, very nice correlations and slopes for the measurements of total lymphocyte counts, CD4 T cell absolute count, as well as CD4 percentages of lymphocytes. And all of this can be obtained using only 10 microliter of blood per sample. The Muse system and assays can also help in other aspects of COVID-19 research, such as in drug repurposing study. And here we show an example of looking at antimalarial drugs. On the left plot, 3 different antimalarials are compared for the ability to reduce Plasmodium falciparum infection or red blood cells using the Muse RBC invasion assay. And you can see the comparative potency of these drugs at the different concentrations. Now, when these same drugs are applied to other cell types, and this you can see on the right, when these same drugs were applied to Jurkat cells and apoptosis was studied using the Muse annexin 5 and dead cell assay, you do notice other effects in that drugs such as dihydroartemisinin, for example, start showing significant apoptosis even at low concentrations of the drugs. And this could be desirable or undesirable depending on your strategies. I'd like to now move on to another of our microcapillary-based platforms, the Guava EasyCyte systems. These systems pack a lot of analytical power in a very compact format. Both single loader systems and which are called the EasyCyte single loader systems and high throughput systems, the HT systems are available, which can do samples in tubes or 96-well formats and still fit into a hood. The systems are based on microcapillary cytometry and enabling small sample sizes and absolute counts and low waste. Several instrument options are available for these platforms, ranging from 1 to 3 lasers and up to 14 detection channels, so enriched multiparametric data can be easily obtained on the system. In addition, Insight software, shown on the right, allows for powerful data comparisons with its easy compensation and analysis features, heat maps that allow for quick identification, and even the generation of IC50 and EC50 curves within the software. The EasyCyte systems thus provide a really nice platform for enriched multiparametric analysis along with the advantages of microcapillary cytometry. So with the Guava EasyCyte systems, the capability to do multiparametric immune cell analysis is enhanced, and one can really do some nice TB and K cell analysis with low sample volumes. So here we show an example of staining CD checks plus and CD checks with low blood controls with a combination of CD45, 348, CD8, 19, and 56 antibodies. Results for the absolute count and percentages of lymphocyte outputs are compared to the vendor-provided limits for the sample on the right. On the plots shown in the right, the upper panel is providing the absolute count comparison, and the lower panel providing the percent-positive cell comparison. And what you can see is that you can get a quick verification of the performance in terms of identifying and correctly and accurately quantifying these populations. And as I mentioned, this kind of immunophenotyping can be obtained with just 10 microliters of whole blood in a no-wash assay with plates or in tubes. We've done more extensive studies where we've applied similar strategies to stain a large number of blood samples, compared results. Here, the blood samples were stained with CD3, 4, and 8, and the results were compared with platforms that utilized IVD methods and reagents. Our data demonstrates very nice correspondence in CD4 absolute counts, determination, showing nice correlation coefficients as well as slopes in the comparison. One can also perform more detailed analysis. Here, for example, the system was utilized for dissecting naive memory T cell subpopulations. This is an 8-color staining performed in a no-wash format with identification of CD4 and CD8 T cells, and then further differentiating them with using CD45 RA and CD62 L markers to identify memory and naive subsets. Again, absolute counts and percentages of these populations can be obtained, and this is very valuable in COVID-19 research. Another area of COVID-19 research is the search for effective antiviral compounds or drugs. Several recent papers have looked into strategies on different cell lines on how known or new antiviral compounds modulate viral activities using cytotoxicity assays. The Guava EasyCyte HT system, with its plate-based screening and multiparametric apoptosis assays and Insight software, can be very valuable in these cytotoxicity studies, and I can show you one such example. So here we show you some examples of a multiparametric assay, which is the mito damage assays. The mito damage assay provides information on 3 parameters simultaneously: mitochondrial potential, annexin 5-based apoptosis, and cell death. The dot plots show you the simultaneous changes in these populations on whether there's both, mitochondrial change and annexin 5, or annexin 5 and cell death. So the plots give you really very detailed, comprehensive information. Below in the 2 graphs, we show you the application of such an assay to treatment with 2 different compounds, etoposide and staurosporin, where Jurkat cells were treated with different doses. And the impact on all 3 parameters with a single assay can be studied. And what we notice in both cases is there's no impact on cell death, but there is significant depolarization and apoptosis taking place. As more significantly in the treatment condition with staurosporin, where you can see both there's a lot of depolarization and annexin-based apoptosis detection. So these kind of assays can thus give highly enriched information to the user in a single experiment in distinguishing impacts. And they may help speed up compound identification or drug repurposing strategies and provide a deeper understanding of the type of impacts in these drug studies. To summarize, COVID-19 research requires the assessment of blood and cellular parameters for understanding disease mechanisms, the immune response, and research on drugs or compounds that modulate viral activity. Here we've shown that the simple low-cost Guava mu cell analyzer provides several 2-color assays for the simplified determination of PBMC concentrations, for the characterization of CD4 T cells, CD8 T cells, and B cells in whole blood or PBMC samples. A range of apoptosis assays on this platform can also provide valuable information in compound or drug studies. The Guava EasyCyte systems, which use microcapillary cytometry, allow for multiparametric tube or plate-based analysis. More complex immunophenotyping data and multiplex apoptosis assays can be easily performed on the system and provide highly enriched information to researchers who are engaged in various dimensions of COVID-19 research. With that, I'd like to now hand this over to my colleague, Dr. Haley Pugsley, for the next part of the presentation.